sensation and perception. Sensation and perception determine how we take in and make sense of the world around us. We're going to focus our discussion on vision because vision is a little more complex than some of the other senses, but if you understand it, then learning about any other senses, if you ended up becoming a psych major, for instance, is relatively easy once you understand how vision works. In general, we want to distinguish between sensation and perception. Sensation is the ability of our sensory organs to pick up information in the form of energy in the environment and transmit that to the brain. So that involves two steps: collecting the data, and for that we need appropriate sensory receptors: eyes, ears, skin that has nerve endings, and so on. So the collecting of that data using our sensory receptors allows the next step to happen. That is the transduction of that information, or the transformation of that energy from the world into a message that your brain understands. Electrochemical energy, which we learned about in the chapter where we talked about neurons and the action potentials. As an example of sensation in the visual world, there is what is known as the electromagnetic spectrum. The electromagnetic spectrum is this long spectrum of different types of light energy that exist in the world. Of all of the energy from gamma rays to AC currents that exists out there, humans can only detect the little portion that looks like a rainbow here in this picture on the right. So this little section right here is the visual information that human eyes can detect. And different animals have different abilities to detect different kinds of visual stimuli. You might think that you live in the quote real world, that what you see when you walk around in the world is reality, when in fact you only see a tiny sliver of what is really out there. There are some animals, including some insects, that see ultraviolet light. Ultraviolet meaning beyond violet, which is the end down here, the lower end of the visible spectrum for humans. So some insects are able to use ultraviolet light to help them find nectar-bearing flowers. There's also some animals that can see infrared. Pit vipers are able to see an infrared glow on warm-blooded creatures, so when they're hunting, it is easier to find their prey. For humans, we just have this section that ranges in wavelength from blue to red. Incidentally, I used to do various experiments on color perception in pigeons, and they have a nearly identical visible spectrum to humans. That is, they see all of the blues and the greens and the oranges and the reds in a way that's very similar to humans. Now, your eyes take in this information, but it has to be converted into a message that your brain understands. Interpreting the message is perception. So in perception, your brain interprets those raw sensations that it has taken in and translates the sights, the sounds, the smells into something meaningful or understandable. Now there's two questions that we can ask when measuring sensation. We're going to ask them first in what we can call English, so to speak, very simple way of stating the questions, and then we'll state them the way that psychologists ask these two questions. So the first question is, what is the least amount of light that can be detected? That is, if something is very, very faint or very far in the distance, what's the least amount that we are able to see as humans? The second question is that given a stimulus that can be easily detected by the human eye, how much brighter does it have to be before you can tell that there's a difference between the original and the new stimulus? So let's look at these two questions a little more closely. This is question 1, now asked in a way that psychologists ask the question. It's a question about your absolute threshold. So for example, for one of your senses to notice something, there has to be enough of that something to detect. That enough is your absolute threshold, the minimum level or the smallest level of intensity of a stimulus that must be present for you to detect it at least 50% of the time. So in a silent room, you would hear, for example, your dog barking, but you might not hear your dog breathing because the sound is not loud enough to detect. Psychologists have measured people's absolute thresholds and discovered that somebody with good 20/20 vision can see a candle flame 30 miles away on a clear, dark night. So if you have a house with a view and somebody 30 miles away on a clear night has a candle in the window, that's visible to humans at least 50% of the time. Now, why 50% of the time? We have good days, bad days, sleepy days where we didn't get enough sleep, or days when you might be hungover or something. And so the idea is that at least half the time you're able to detect these stimuli. For hearing, the absolute threshold is a sound of, say, a ticking watch 20 feet away under quiet conditions. You can detect one drop of perfume diffused throughout 3 rooms. And taste, if I put a teaspoon of sugar into 2 gallons of water and shook it up, you'd be able to tell that the water was sweet. 1/2 a teaspoon in that same 2 gallons of water, and you wouldn't notice most of the time. For touch, you can feel the wing of a fly falling on your neck from a distance of 1 centimeter. So these are absolute thresholds, what humans are able to detect. Obviously, some animals can detect things that are harder for humans to see or hear or smell, and so on. For instance, dogs obviously have a very powerful sense of smell. They can tell that you have been in a particular room hours ago and what direction you were walking as you went through the room based on the smell. Now, the second question is a question about difference thresholds, or the just noticeable difference. That is, how much of a stimulus change is necessary for detection. And there is a basic law about this, and there's not very many laws in science. Laws are theories that have graduated to a higher level because they've been demonstrated to be correct more often than not. So the just noticeable difference increases with the magnitude of a stimulus. What we mean by all of this is that the larger the initial stimulus, the larger the change has to be for you to detect a difference. So if I handed you an envelope and said, here's a little envelope, the size that you might mail a check to somebody in the mail, not that most of us even write checks or send mail all that often anymore, we're a digital world, but imagine I handed you an envelope and asked you to estimate how heavy it was. And then I instead gave you a envelope with a quarter in it and said, now, do you notice that it is now heavier? It would be pretty easy to detect that the envelope with a quarter in it is heavier than an envelope with nothing in it. However, if I took your backpack, which is probably filled with books and notebooks and devices, possibly a laptop or an iPad, and I tossed a quarter in it when you weren't looking, would you pick up your backpack and go, hey, who's been messing with my bag? It's suddenly that much heavier. No, you wouldn't notice. The reason is because your backpack was so heavy to begin with, I would need to put something in it that is really much heavier than a quarter for you to notice the change. So a book or a brick might get you to notice the difference. And this is Weber's law. The just noticeable difference is a constant proportion of the original stimulus intensity. That means big stimuli need big changes, small stimuli need small changes to be noticed. Another place where it applies is in all kinds of sales or in discussions of salary. So, as an example, there's a lot of stores where you might go in and maybe you're buying a pair of jeans, and when you get to the counter, they ask you if you'd like some socks to go with your jeans. The reason is that the jeans might cost $100, let's say, and if everybody who was buying jeans added on a $6 pair of socks, they make that much more money, and the person who's now spending $106 instead of $100 doesn't really notice a huge difference between those two things. Now, you can only do it that one direction. You can't go in to buy a pair of socks and have them say, you know what would go great with these socks? Maybe some jeans and a leather jacket. Because now you're asking somebody who was going to spend $6 to maybe spend $300 or something. So this is a very important concept for people who are in business, or if you are negotiating your own salary. Once you graduate from college and you get your first, quote, real job, you need to ask for the highest salary that you are worth in terms of your education and your experience. Now, some people think, I don't want to be too greedy because I'm getting a job offer and I should just be happy. But once they set your starting salary, every raise will be keyed to the starting salary. So if you settle for less, thinking you'll be able to ask for more later, you will always be a bit behind. So Weber's law is a very powerful concept for people studying things like business or economics. Now, because I said we're going to focus on vision, we're going to take a little tour of the human eye. And we're looking at an eye that is looking straight ahead, and a moth is in its visual field, so it's staring at a moth. And what you can see here, this is the front of the eye, the clear part, or the cornea. And light passes through the cornea, through the pupil, and the pupil is basically a hole in your eye. It's what you think of as the black spot, but there's really not a spot there. It's actually just a hole. And that pupil is surrounded by muscles. These two pieces right here that I'm highlighting with my arrow are the iris, and the iris is what you think of as the colored part of your eye. Mine are green, yours might be green or blue or brown or some combination thereof. Basically, they're striated, they have little stripes in them because they're muscle tissue, and they control how wide or how narrow the pupil is. So if I shined a bright light in your eye, the pupil would contract and get smaller. And if I put you in a dark room, it would expand, and that expansion and retraction is helped out by the iris. Once the light travels through the pupil, it goes through the lens of the eye. And the lens of an eye is just like the lens in anything else that has a lens, a camera, a projector, something like that. It inverts the image onto the back of the eye. And the image then is upside down. So you see that the moth at the back of the eye is upside down. Now, of course, when we look at objects in the world, we see them right side up. That's because our brain puts our eyes, or our images, right side up. And I've posted a little video showing what happens in a crazy experiment that was done many years ago, where a lady put on these special glasses that caused her to make yet one more inversion of the image. So she lived in an upside down world for, I think it was about a week. And you can see some of the things she did at first took an enormous struggle for her to make sense of just pouring a cup of tea, for instance. And then by the end of the video, she's doing something with these goggles on that would make me too nervous to actually do what she's doing. So it's kind of a fun little experiment that you can watch. If you'd like to stop the lecture here and watch that now, you can, or you can come back to it in a little while. So information travels to the back of the eye where you have all of your photoreceptors. And the back of the eye here is called the retina. And in the retina is where you have cells that will transduce the light energy into a neural code that your brain can make sense of. So the transducers are in the back of the eye. Now, we will mention this fovea area. It's the very center portion of that retina. And you see that the retina is kind of a backward C shape. And all of the cells that turn the light into neural code are in this particular layer of the eye. And all of their axons leave the eye out through the optic nerve to head towards the brain. So we're going to look at those receptors that are in this backward C-shaped structure. And they're going to be called rods and cones. Now, at the very outermost portion of the eye, the periphery, which is out here and here, you have only rods. You do not have any cones at the outermost portions of the retina. In the fovea, you have only cones, and it varies kind of proportionally throughout. So kind of here, there's quite a bit of cones with a few rods, a few more rods, and a few less cones. Out here, lots and lots of rods. So we'll see the importance of these two different types of photoreceptors. So your rods are sensitive to light, but not color. They only see in black and white. And they're useful in low illumination. So when the lights are out, you get up in the middle of the night to go to the bathroom and you don't see super well, you're using primarily your rods. And humans have 120 million of them roughly. Now, we also have another light-sensitive photoreceptor called cones, and they respond to color. So the rods are the structures that are shaped basically like rods, and the cones are the structures that have more of a conical shape like a cone. And humans have about 6 million cones, and we use them primarily in good lighting conditions to discriminate colors. So just to give you a few examples, I used to work with both rats and pigeons in graduate school, doing different kinds of experiments with each species. When I was studying color vision, I had to use pigeons because rats don't see colors. They only see black, white, and shades of gray. The reason is that rats and most rodents only have rods in their eyes. They do not have cones. So they don't see color. But the reason they have all the rods in their eyes is that they hunt at night in very low lighting conditions where discriminating colors is not really very helpful or useful in terms of them seeking out food and so on. Alternatively, pigeons have virtually all cones and no rods. So it's kind of funny, but I had a lot of assistants that used to help out in the lab. So when I was a graduate student, my assistants were undergraduate students who were typically psych majors trying to do some research to help get into graduate school. And they would shuttle the animals from their cages, their home cages, to their experiment. And every once in a while, a pigeon would get out of somebody's hands and fly off. And I've had more than my share of students who were freaked out that they had a pigeon that was flying loose in the lab. And it's actually very easy to catch a loose pigeon. You just have to memorize where the pigeon has landed and then shut the lights out. Then you walk, using your sense of touch, you walk to the spot where the pigeon landed and you grab him and then go turn the lights back on. The reason is that because pigeons are all cones, when the lights go out, the pigeon basically goes out. He roosts when it turns dark. So they're pretty much all color vision. Now, I pointed out the fovea, which is densely populated with cones, and it's vital to really good detailed vision. That's where we get our best vision, is when we line up something we're looking at so that it hits that area of the back of the eye. Now, I also pointed out the blind spot where the optic nerve leaves your eye. And at that portion where the optic nerve leaves the eye, you have no photoreceptor. So if light hits there, you don't see anything. And you can find your own blind spot using something like this diagram. You can draw it on a piece of paper, or you could try it with the screen. If you look at the dot with one eye, so open one eye, close one eye, look at the dot and move your head forward and backward. And there's a point at which the plus sign will disappear from view. And that is where your blind spot is. Now we're looking at the top of the brain. Here's the front of the brain, the frontal lobes. Here's the occipital lobes, which you remember from the chapter on biopsych, is where primary visual processing occurs. So we have to get information from the eyes to the back of the brain so that we can start to make sense of what we're looking at. So the information travels through the optic nerve to what is called the optic chiasm. This is where half of the fibers from each eye cross to the other side of the brain, because you remember the contralateral wiring that we talked about in the chapter on biological psych. That information from the right visual field is going to end up in the left hemisphere, and information from the left visual field will end up in the right hemisphere. And they'll do that by half of the fibers crossing to the other side of the brain and half of them staying on the same side. So once information gets to the occipital lobe, we start doing some really detailed processing on what we're seeing. Now we turn from sensation to perception. Sensation involves only the detection of available information in the environment, the sights, the sounds, the smells. What happens to that information next is perception, making sense out of those raw sensations. The raw sensations are biologically driven. They're determined by the sensory receptors that you have. For instance, you and I, as well as most of our family pets, such as dogs and cats, have eyes that are at the front of their head. And because of how they're positioned, we can see depth and 3 dimensions. Animals with eyes on the sides of their heads don't have that information. And we'll look at a few concepts that explain why. But for now, you need to know that perception involves so much more than just pure biology. Perception is really making sense out of information using all of your knowledge, your experience, and all of your previous learning. So for instance, in the famous dual image to the right, there is a picture that you can look at. And if I asked you, what do you see? I'll give you a minute to take a look at that picture and see what appears in the picture. Now, some students will look at this and immediately see an old woman. Others will look at this picture and immediately see a young woman. Some people will easily be able to see both, and others will say, I don't see how you could possibly say there is an old or a young woman in this picture, when there is in fact both. It just depends on your perception, in what features of the picture you're zeroing in on. And some of that may involve your history or experience. For instance, if your grandmother is suffering from Alzheimer's disease, you may be more inclined to see the old woman because your grandmother is frequently on your mind. So if you weren't able to see both of those, I will say that the young woman's face is here with her forehead, her nose right here, her chin, young woman's chin. This is the hair, her ear, and she's wearing a necklace that's choker style. Now, if you wanted to see the old woman instead, this is her very large prominent nose with a wart on the end of it. This is her mouth, and this is her chin, and these are wrinkles on the side of her face. Whether you saw the young or the old woman is heavily influenced by all of the things that bring meaning to your life. So we have to take a look at several different ways in which we perceive information. We look at the world and we see color, we see form, we see distance, and all of those cues help us navigate in the world. So we're going to first ask the question of how do we see things in color? And there's a couple of theories that are in your book. One is called the trichromatic theory of color vision, and one is called the opponent process theory of vision. And let's just cut through all the nonsense and get straight to the answer. Both of those theories are correct. It just depends on whether you are in the eye and trying to explain how the eye itself sees color, or if you're going higher in the brain at layers of processing that occur, say, in the cortex or in other parts of the brain. So let's look at each of these theories in turn, starting with trichromatic theory. Tri means three, and chromatic, chrome is color, so the three color theory is essentially what that means. And what that amounts to is the idea that we have, in the human retina, we have three types of cones: red, green, and blue cones that see essentially red, green, and blue colors, although it's not really literally those colors, but rather wavelengths. So the blue cones see all of the wavelengths at the lower end of our visible spectrum, somewhere in the area of 400 nanometers at the far left, where violet and blue light exists. And green cones are somewhere in the middle of that spectrum, and red cones are at the far end. So our ability to see color, at least at the level of cones, involves how much activity is happening in each of these types of cones. So if red is visible, then your red cones are doing a lot of firing. The problem is that this theory does not explain color blindness. For instance, some people have red-green color blindness, where they cannot distinguish between the colors red or green, or the various wavelengths of those colors. They all look like more or less the same shade. And others have blue-yellow color blindness, where they can't discriminate between blue and yellow. And this theory of color vision doesn't really explain that. So we have to look at another theory of color vision in order to understand why that occurs. So I'm going to let you stare at this picture of this flag. It's the American flag, of course, but in strange colors that we're not used to. What I'd like you to do is spend about 60 seconds or even more, 60 seconds to maybe 120 seconds, so 1 minute to 2 minutes, staring at the white cutout portion in the middle of this picture. And when you've stared at it for a while, and you don't get bored and you don't look away, but you stare at it for long enough, then what you should do is turn your eyes to look at a blank wall. A white wall would be perfect, or a light colored wall, and see what you see. So if you'd like to try this little experiment, I promise you that the result of it is going to be very cool. And you might want to pause this lecture so that you're able to stop here on this slide and stare at this picture for just a moment. And then again, after staring at that little white spot in the middle for 1 or 2 minutes. You look over at a white wall or something like that. If your flooring is light or your walls are light, look there and then see what happens. So what you probably saw if you did the experiment in the previous slide was an after image that looked like the American flag in the way that we're used to seeing it, in all of its red, white, and blue glory. The reason that you saw that is that the colors of our cones are actually hooked up as complementary opposites. So red-green is hooked up together, and blue-yellow is hooked up together, and of course black and white are hooked up together. So you stared at the screen for a while and were seeing green and yellow and black, and you fatigued those receptors. So when you looked at the white wall next to you, the paired opposite to each of those fired, and that's why you saw red, white, and blue, the paired opposites to each of those colors. So the opponent process theory can explain these color after images, and it can also explain why certain people have color blindness, in the sense that you will get blue and yellow confused or red and green confused if you have one of those specific types of color blindness. Now the next question is, how do we recognize forms? That is, how do we recognize patterns or objects? Like if you graduated from high school a year ago or a semester ago or 10 years ago, and you see somebody you went to high school with, how is it that you're still able to recognize them? There's a group of principles that helps to explain how we understand the kinds of objects that we see and make sense of them. And they're called the Gestalt principles of perception. They're a series of principles that describes how we organize bits and pieces of information into meaningful wholes. In fact, sort of a catchphrase, a slogan, if you will, for Gestalt psychologists is that the whole is worth more than the sum of the parts. And it comes from the concept of the Gestalt, which is a German word for pattern or whole or form. So what are some of these Gestalt principles? Well, your book looks at proximity. That is, the closer that objects are to each other, the more likely they belong together. So if you walked into a restaurant with your best friend, prepared to get a table for two, and somebody walked in right behind you and stood very close to you, and they were waiting for the rest of their party to show up, let's say they had a party of 4, when the hostess sees the 3 of you standing together, they're going to assume that you're all together and might say, "Would you like a table for 3?" And you'll look at them and say, "What are you talking about?" "No, we would like a table for 2. The reason is, of course, this proximity issue. When you see things together, you assume they go together. The other concept that is interesting in the Gestalt view of object perception is similarity. That is, if objects are the same size or color or orientation, that is, they're facing the same direction or the same distance from you, then that's probably a group of objects or a pattern of objects and not just a bunch of random things that just happened to be similar. So if you walked into a room with 10 of your bridesmaids and you are the female, you're the bride in this scenario, you're standing there with a bunch of other females, you're all about the same age, you're all reasonably attractive, and standing nearby is your one male bridesmaid, if you will. Maybe one of your best friends happens to be a male, and that would confuse a hostess as well, that you wanted a table for all these women and the one man. So we tend to see all of the women as going together, and the one guy maybe is not part of the group. Another concept is the closure concept. That is, if one part is missing in an otherwise complete shape, you're likely to overlook the missing part and assume that all of the parts make a whole. So this, with the series of little black, sort of almost like Pac-Man faces with a triangle in the middle, you see a triangle that might be sitting in front of all of those three black objects, which you assume to be circles, even though you can't see the edges of the circle. Also, the figure ground concept. In a way, the old woman, young woman that I showed you earlier in this lecture is another example of this figure ground concept. That is the idea that the eye differentiates between an object and its surroundings. And sometimes you have to make guesses about what is the foreground or the key item and what's background information. So if you see your friend standing in front of a wall that is covered in wallpaper, your friend could be the figure and the wallpaper the ground in that particular context. Another important question is, how do we judge the distance of an object or the depth of an object? How far away is an object to us? And understanding this will help us understand a couple of perceptual illusions or visual illusions that we have when we see things and misperceive them. So there's two ways in which we can tell whether something is near or far from us. And one of those ways is using what are called monocular cues, and the other way is using binocular cues. Now, mono obviously means one, and bi means two, so ocular is in reference to vision. So using one eye or using two eyes. And the monocular cues to distance are things like linear perspective. That is, if you were driving on a long straightaway for miles and miles and miles, in the distance, the lines of the road in front of you converge. So have you ever been taking a long drive, maybe out to the desert or to Vegas, and you're on a long stretch of highway, and you think to yourself, I need to turn back because miles ahead, the road is too narrow for my car to get through. Okay, that never happens because we understand that the reason that the road looks narrow in the distance is that that is further away. Another monocular cue is relative size. That is, things are close to us tend to appear larger to us in our brain. And so we assume that the thing that is larger is just closer to us. And we're going to see an example of that when we look at some perceptual constancies as well. Another example would be a texture gradient. That is, things that are close to us appear detailed in texture, and things that are far away seem sort of blurred or blended. Another way we can detect depth or distance is with interposition. That is, let's say I were standing in front of a classroom filled with 150 students. I would be able to tell that the person in the front row is closer to me than the person 3 rows down because of how they're positioned one in front of the other. And the person in the front row, I can see them from head to toe. And the person in the third row, I can probably only see them from the waist or chest up. And I can use those cues to help me understand what's in front and what's further behind. Now, we can also use binocular cues, which means both eyes working together. And I hinted at that when I mentioned that certain animals cannot use those cues because of how their eyes are positioned. But because our eyes are in front of our head, the physics of our visual system, or the optics, means that we can rely on a couple of tools that involve cooperation between both eyes. And the binocular cues include convergence. That is, let's say that you had a bee in the room. You're in a room and there's a bee. I don't know how you would feel, but I'm allergic to bees, so I'm not too thrilled when there is a bee in the room. Actually, it's more terrifying if it's in your car because you're kind of stuck in there. And how terrified? How panicked do you need to be? Well, if your eyes are kind of relaxed and the bee is far away, you can see the bee at the very back of the room, your eyes are going to be relaxed and they're not going to be crossed. They're going to be sort of physically positioned, sort of at the edges of their sockets. But if the bee starts to fly into a place that is just in front of your nose and you are watching it, your eyes are going to start to cross. And that is convergence. So you may have been to an eye doctor and had them test you where they take something like a finger or a pen and they move it towards your nose and they ask you to keep staring at it. And you might have been a little self-conscious because you knew you were crossing your eyes and probably looking a little bit silly, except for the fact that they do that 24/7, 365 days of the year. So they're not very affected by that. But anyway, that's the concept of convergence. And the concept of retinal disparity, in order to understand it, if you hold your finger at arm's length out in front of you and then close one eye and look at your finger, which is pointing up, and then close that eye that was opened and look at your finger with the other eye, and then close that and open the other eye and close that and open. Do that a few times. You're going to see your finger bounce around a little bit. The view of your finger from each eye is slightly different. That difference is retinal disparity, and your brain uses that to help you make sense of depth or distance. Now, there are also some perceptual constancies. They include things like size, shape, brightness, or color. In the case of shape, if you look at those three doors that are up there in the right-hand side of the screen, we understand that all of those are doors. They're all rectangles. They're all the same shape. They're just, one is closed, one is partially opened, and one is about halfway opened. But we don't see those as different types of shapes. We still see them as rectangles. So for size constancy, that's where we recognize that an object remains the same size even though the image on your retina of the object changes as it moves away from you. So as an object moves off into the distance, it starts to look smaller. And that informs our perception of size. So when you're flying in an airplane, you know that just because houses and cars look like ants to you, you still realize that they are in fact full size. For color constancy, that's the recognition that an object is the same color even though different amounts of light fall on it. For instance, an apple looks red whether you're having it out at a picnic on a bright summer day or in a dark room in a classroom, let's say, while you're watching a movie. In fact, the idea of color constancy kind of broke the internet a few years ago. If you remember that dress that some people saw as black and blue, which was in fact its true color, and others saw it as white and yellow, and that was a misperception. If you saw the dress as white and yellow, it was in fact blue and black. And part of that misperception for some people is the fact that the photo was not a really great photo. It was just a candid photo of somebody who was at some sort of wedding or a party and had their photo taken and posted it online and had different friends commenting on their lovely black and blue dress or their lovely white and yellow dress, which they found very confusing. Now, in a sense, when we see something that looks ambiguous, we have to make guesses about the context of the information. So for the people that are seeing that as yellow and white, they're making some sort of assumptions about background illumination. Maybe they've misperceived it as being in shadow, and therefore they see the dress as white and yellow. I don't think neuroscientists even fully understand what makes some people see that one way and others see it the other way. But it's clearly a fascinating subject that just about everybody on social media happened to share at the time. Now, some of those cues that we use for distance or for size and our perceptual constancies can sometimes, as in the case of that dual colored dress, lead us to misperceive information. So there's some famous visual illusions, including the Müller-Lyer illusion, which is portrayed here in this slide. You see two lines, A and B. Which of those two lines do you think is longer than the other? Now, if you've read ahead, you know that these two lines are the exact same length. I know they're the exact same length, and I've taken advanced psychology of vision. I took a psychobiology of vision class in graduate school and a bunch of different classes and understand what's going on from the nervous system all the way up. And I still feel like the line at the bottom looks larger, even though I know that they're the same size. And if I asked you why, you might say it has something to do with the direction of those arrows, and you'd be correct, but it's not the full explanation because we need to know why it matters that those arrows are going different directions and what it is doing to our perception. And part of it is, again, learned experience. The A example looks like the edge of a wall that is jutting out at us. So if you imagine this is a corner wall in your room where this wall is sort of sharp, you can run your finger along the edge here. And this is a wall that's receding into the distance. So if you look around your house, you can find a wall that is pointing out at you, a corner, and you can find a wall that is jutting away from you. And because we typically live in homes with square rooms, this illusion affects just about all of us. If you grew up in some Aboriginal tribe that only lived in round huts, then you wouldn't fall for this illusion. Another illusion that's somewhat similar is the Ponzo illusion. That's where the top line looks much longer than the bottom line, even though they're the same length. This is due to our perceptual constancies. We assume that things out in the distance are going to appear smaller to us than if they were close to us. And because of this linear perspective and those lines sort of converging in the distance, we assume the back line, that pink line at the back, is further away from us. And if it looks the same size while being that far away, our brain assumes that it must be huge. And that's why it appears to be larger than the one that appears to be closer to us. So both the Müller-Lyer illusion and the Ponzo illusion involve misinterpretations of both linear perspective and size constancy. And so again, this just illustrates the concept that we started out with when we started talking about perception. That is, your experience, the context, the background, the surrounding information, all of those influence what you see. That's it for today.